This program is a presentation of UCTV for educational and non-commercial use only. Check out our YouTube original channel, UCTV Prime, at youtube.com/UCTV Prime. Subscribe today to get new programs every week.
Funding for this program was provided by the UCLA Office of Instructional Development.
These are the prairies of central Nebraska, where my ancestors got off the train to homestead and started their search for a better future. As a result of that, I've always been interested in my ancestors, and I think everyone in the world, especially Americans, are interested in where we've come from. But we're also interested in knowing where earlier ancestors came from and where they came from. We're interested in knowing the origins of humans. We'd like to know how are we related to Neanderthals. And furthermore, we'd like to know how we're related to other organisms, to even deeper organisms. So first I wanted to tell you a couple of things about evolution and talk to you about well, the curiosity, where we came from, and then how to improve human health. What do earlier organisms tell us about improving health? Well, for example, if we can find the early ancestors, they tell us about signaling pathways. But we must get the correct trees. In other words, we have to get the correct pathways to make sense of everything that's known from the biology. Similarly, they promise to tell us how to design new bioenergy mechanisms. The mechanism of photosynthesis has absolutely transformed the Earth, and I'll talk to you about that later. But if we know the steps in which that happened, then we also know how to put all of the pieces together so that we can start to understand photosynthesis and devise new biofuels. Now, Charles Darwin started evolution off, he and Alfred Wallace, with the bifurcating tree. A bifurcating tree starts from one organism to make 2, and 2 to 4, and 4 to 8, and so forth. Meanwhile, these organisms start to compete with each other. Okay? So he liked to call it the survival of the fittest. And that's an interesting metaphor that's existed. But there's another type of evolution that we need to know about and that's starting to become more important. And that is convergences, where two organisms come together to make one. And of course, in a sense, we're an example of a convergence. I mean, we had the sperm from our father and the egg from our mother, and we're the descendants, so we really have to have genomes coming together to make a new type of individual. But this is done time and time again in evolution, but it's not studied. The reason is that it's very complex to do convergences and tree-like at the same time. In fact, if you think about it, the tree of humans is a tree, but of course it's not a tree. If anybody's interested in genealogy, you realize they don't draw trees, they draw all the parents and they do this, and they get into a far more complicated arrangement. So the question becomes, how do we go about this, and what's triggering this interest? It turns out that evolutionary biology is really in transition. We were sure, absolutely sure, that when we got the first genomes, that these would tell us about, do the tree of life, we'd have every little bit of life understood, there'd be no problem. It didn't happen. We, in fact, began to wonder, and different people proposed, well, we have horizontal gene transfers, and of course that was part of what our lab proposed as well, that individuals, when they live together, they start to exchange genes, so that there's a really complex arrangement of gene changes. That's, in fact, when we looked at the genes from various genomes, we find out there's no single tree, that every gene tells a different story. It's turning out that we can start to derive rings, and I'll talk to you about this ring of life, but I want to first of all tell you about the organisms that make up life. In general, there are two types of organisms. They're prokaryotes and eukaryotes. Prokaryotes are the organisms that are present earliest in the fossil record. They're very small, single-cell organisms. They don't have a nucleus. They can photosynthesize. They were on the Earth, as people like Bill Schaap from UCLA have shown, for very long periods of life, and they are extremely important. They have changed the way the Earth is. The eukaryotes are like us, like Chancellor Bloch referred to. We're different, you know. We have big cells, we have a nucleus, and in addition to that, we have organelles, like the mitochondrion and the chloroplast. Now, the chloroplast is the organism shown in these is the organelle shown in these slides. It's in red. These are plant cells. And these chloroplasts are busy taking the light in, and they make food for the cell from the sunlight. It's absolutely remarkable, because without plants, we would never have had animals, because animals have to eat this food that's made from the sunlight, and none of us would be here. So thank a plant today. Anyway, eukaryotes, like us, also have organelles, because we learned a trick. Well, we didn't learn it. We stole it from the prokaryotes, the trick of how to breathe oxygen and how to thereby enable us to get more than, maybe 10 times more energy out of the foods that we eat. So this happens to be the two tails of a bat sperm. So this is one single cell. Now, the bat sperm has one main thing to do, and it is to move as fast as it can to try to find the egg and against all the competition and fertilize the egg. So these little things in it, and remember, this is all just one cell, are mitochondria. So they're making the energy, and if you wanted to go the fastest, you've got to make the most fuel. And that's why they've evolved to just be totally filled with mitochondria to succeed. Okay, now the question, I've been talking about trees and graphs, but I haven't told you how to derive them. And I wanted to, a lot of this is intuitive. If you think, for example, of cats and lions and dogs, you can say, well, gee, a cat and a lion, they have a lot in common. They have similar teeth, they have a similar skull. And we could say, and that would be the phenotype of the animal. In some ways, the cat is more similar to the lion than either the cat is to the dog or the lion is to the dog. And that's the way we can use phenotypes. Genotypes now give you the same sorts of results sometimes, but they may not have quite the same numbers, might not be the same lengths of the branches, but they are usually, they can be compatible, but they contain a lot more information, so they're much more reliable. Again, if we had gene evolution, then we have long genes, thousands of nucleotides. And the nucleotides are the bases in the RNA, and they have 4 different names, A, C, G, and T. And we can compare, say, the cat sequence, which is the order of these nucleotides on a gene in the cat, with that in the lion. and we'll find there are a few changes, but they're a lot similar. It's these changes and similarities that guide us, and we can count the number. We can actually do sort of bean counting, in which we say, how many times do I have changes versus how many times do I have the same there. Similarly, you can do something like that with genome evolution, except instead of having thousands for a gene, you can have thousands, tens of thousands of genes, and you can treat them, is this gene present in the cat, for example? Is it present in the lion? Now, we don't, and we can look for differences, just like we did over here. Are they absent in one, present in the other, present in both, present in one, absent in the other? We can do all of these things, and we can also build trees, but we can also build graphs. And that makes the gene absences and presences more powerful. Furthermore, we can get further back in evolution, because our goal is, you know, always to go push the envelope, try to go back as far as we can. So if genes change very fast, you know, they can change in, a position can change in 100 or 1,000 years, but proteins are gained and lost very slowly, maybe in the millions or tens of million years. Now, the slower something changes, the further back in the past it can take you. And that's what we want to do. So we have a problem, though, and that is about absences. What does it mean to say that a gene is absent in both of something? We don't even know what gene it is. So how can we say which gene is absent? It's sort of like having a glass in front of you, where you ask, is this an empty glass of wine, or is this an empty glass of water? And looking at this group, I have a feeling what it'll be after the reception, okay? So anyway, I mentioned bean counting, so I thought I would show you an example from Cambridge, where Chancellor Bloch said I'm a former overseas fellow. And when you go to high table, and that's sort of this little table you sit at, and you wear gowns and all of this stuff, you sign up before and you take a guess. So if Laura and I were to go, we would, you know, we might take 2 guests, we'd sign up 4. But they maybe have 20 pages in this list, and people can't take the time to go through, and is there one here, and they do the addition, and they get it all mixed up. So you're also, if it's Laura and me and 2 guests, we would take 4 beans, we would put the beans in the carafe, and just about 1 o'clock in the afternoon, when they start preparing the dinner, they'll come, they'll count the beans. They don't have to worry about who's there. They just want to know how many beans and how many dinners to prepare. So that's the origin of bean counting. These are our kids, Jeremy and Caroline, when they were young. And this sort of got me out of the field of ribosome structure. I mean, it was such I did it for several reasons. One was Bill Schopp founding WEEG here at the Wednesday Evening Evolution Group, but also I kept getting more and more interested in evolution. And we used to go to these petting ponds at aquaria. And one day, you know, they have starfish and things that won't hurt them if you examine them. And one day, I decided that I wanted to, they said, this is a sea hare. And I said, I don't know what a sea hare is. What could that possibly be? So I went back to UCLA, and I got a book, started reading about it, and it turns out its scientific name is aplysia. If they had said aplysia, then I would have known it, because that's an organism with the big nerves. But I looked further, and I realized we don't know how it's related to other things very well. In fact, the more I looked, I realized we didn't know anything. So our lab had been very busy sequencing ribosomal RNAs, and I said, you know, none of these plants have been ever sequenced, or not plants, these animals have ever been sequenced, and we ought to start trying this. So we started doing it. And that's how we developed, ultimately, the new animal phylogeny. I'll tell you the first part of it. It says that the starfish, these sort of 5 symmetry-fold organisms, are more closely related to the chordates, those animals with the spinal cord. And over on the other side, that the arthropods, which are things like insects and and crabs and so forth are more closely related to the mollusks, which are clams and all of their like. So these are the bilateral animals. So bilateral animals are the ones that have a left and right side, and we're approximately symmetric. Now, I wanted to tell you a little bit about some of these animals because they are really incredible. This is a tardigrade, and it is just barely visible with the eye, with the naked eye. You can, if you want to see some, you can go up on the coast and take a little moss off the tree, shake them out, and there'll be some things you can put them on a white paper and you just barely see them. Most of them are in a dormant phase. So you take them back to the lab and you add them to a little drop of water. And they'll, on a microscope slide so you can watch them, and they'll be down at the bottom, and they'll start, in about a minute or two, they'll start walking along the bottom of the slide. I mean, it's just like adding, instant coffee in insects. So you add it, and you go on, and they are the most amazing creatures because of this ability to dry out, desiccate. They've been taken into space, and they've been put out in the vacuum of space and left for several weeks. They take all the radiation, everything, take them back to Earth, put them in a drop of water. In a minute or two, they start walking around. They're just fine. They can reproduce everything. So the record that I know was a museum specimen, which was a 100 years old bit of moss. And somebody said, let's see if there are any tardigrades in here. They flicked it, took out the tardigrades, and added water, and 2 minutes later they started walking, and 2 minutes later after that they died. But nevertheless, but nevertheless, they walked. So that is one hardy animal. Okay, now another type of animal that I'm going to mention are the phoronids. This is a wonderful picture taken by a former colleague from Shiprock, California. Now, Shiprock is, it's west of Catalina, but these are beautiful phoronids in the background. I don't have any close-ups of them. It's taken by Jim Moran, a former UCLA faculty member. And, but this is a bryozoan. It's very similar. They have these tentacles, which are called lophophores, and they filter feed. They feed on other very tiny eukaryotes, so forth, and grow that way. Now, in the tree of life, the old view, they were close to the echinoderms and the chordates. So we thought that they were good models to use for human evolution. The distant models, because if we go down here, hopefully we can find out something about signaling pathways, their evolution. Turned out they aren't. We got the sequences, and immediately they popped over here into a new group. They were closer to the worms, to the annelids. Now that's because, I think, by their form, they're so different that you can't really compare them with anything. And that's many of the changes involve that kind of change. Similarly, we had the nematode worms, which are over here. We used to think they were good models for example, things like longevity, because you starve them and they live far longer. And we said, well, this really applied to our closest relatives, the humans up here in the deuterostomes. But we now know they are more closely related to the arthropods and the nematodes. And you ask, well, what do they have in common? It turns out that they both molt. Now, growing up in Nebraska, we had lots of insects, lots of grasshoppers. You'd see these little grasshopper shells from when the animals had gotten too big for their exoskeleton. They broke it, and then they'd go and they'd secrete. They'd be vulnerable to predators for a day or so, and then they'd secrete a new exoskeleton and go on with the process. That was a very important thing, because the most speciose animal group we have is insects. This adaptation was absolutely remarkable. But it did show that molting helped us clean up and understand better where our ancestors are, our closest ancestors. So that's what I mean when I say evolution is an important framework for understanding evolution. Now, as Chancellor Block mentioned, that's what I won the Darwin Wallace Medal for this May, and I wanted to tell you just a little tiny bit about that because it's such an amazing place. It's where Darwin and Wallace first presented their paper in 1858. And so you go into the place, and on the far side, you see this portrait by John Collier of Darwin, very famous, but the eyes are just crazy. You know, you walk in, and of course he's sort of looking over here, and back behind me would be on the other side a portrait of Wallace. So they're kind of looking at each other, but whoever goes in says they always feel that the eyes of Darwin are watching them. So I don't know about, I do know that it's a weird feeling. It's a wonderful feeling too. So now let's talk about plants, because I told you they were important. We wouldn't be here as animals without plants. This is a cover from Nature, the scientific journal. And if you look at it carefully, I love this cover. This is a DNA helix, but it's made of potato chips, and then the bases linking them are of French fries. So that's inventive science by itself. I mean, you need something like that to get published in Nature. Anyway, the potato genome, this only came out about 4 weeks ago, but it's extremely interesting. I think it's interesting about what we say as in our perceptions as being sort of at the top of the feeding order in the world and on Earth. Because we think we're so smart and so bright, and we have this big brain. And, you know, we thought for sure when the human genome came out that we would have the biggest animal brain, but we don't. I mean, all animals have pretty much the same number of genes in our brain. I meant number of genes, I didn't mean brain. So we end up all animals, but surely animals have to be a lot smarter than plants. This just cannot be. Well, it turns out in the genome, plants have about 40%, 45% more genes than humans. So we have roughly 23,000, and they have 34, 35,000. So I guess the take-home lesson is, next time somebody calls you a potato head, take that as a compliment. Now, I like quotes, and I like, you know, a lot of people give talks, famous speakers like Descartes and so forth, or famous people. And so here's my famous quote for the next subject. It is, in my career, I found that thinking outside the box works better if I know what's inside the box. Okay. It only has one drawback. The author isn't so well known. Starbucks coffee quote number 153. Anyway, but we do, in talking about prokaryotes, we want to know about what's in the box. And I'll tell you a little bit about their diversity now. This is, at least the people in the first couple of rows can see this, but these are lots of people's faces. And human eye is very good at picking out different faces. But if you look at them carefully, you'll realize it's just 4 people in different poses, different smiles, all sorts of things. Now, our problem with reconstructing prokaryotic life has to do with the horizontal gene transfer. We want to minimize that process. So we've made as big groups as possible of the prokaryotes because, remember I said like organisms, related organisms prefer to transfer genes with each other. So if we get big groups, then sort of like in this picture, everything fits within the square with maybe a little exception. And think of those as the rare transfers between groups. So it reduces that effect. It means now we can start to look at what are the big groups. One group are the methanogens. And the methanogens consist of organisms that eat carbon sources, but they breathe hydrogen, so they make methane. In fact, when you turn on the kitchen stove, if you don't have an electric one, if you have a gas stove or a gas furnace, you're burning methane probably produced by methanogens in the past. Another group are the double membrane prokaryotes. We've known about these for 125 years. They were first called the gram-negative prokaryotes, identified by their by how they stained for light microscopy. And these include things like the cyanobacteria. There are a lot of photosynthetic groups. In fact, almost every single photosynthetic bacterium, except for one little organism, is found in this group. So there's something about the double membrane arrangement that helps photosynthesis. But that includes the cyanobacteria, and I'll come back to the cyanobacteria in just a minute because they're so important. Another group includes the firmicutes. Now, the best known one would be anthrax after 9/11, because it's a bacterium that makes anthrax. It has a very interesting history. But also it includes Clostridia, which are fermenting organisms. And within the Clostridia, there is one photosynthesizer. And then we have the actinobacteria. Actinobacteria sometimes form these complicated structures of multi-cells, but they aren't really quite multicellular. They include also parasites like the one that Mycobacterium tuberculosis causes tuberculosis, or a bacterium that causes leprosy. Those are important. Those four represent pretty much the diversity of prokaryotic life. Now, we use the presences and absences to start to look and construct a tree of just these four. But it turned out to be more complicated than we had thought, because the double membranes have two origins. They, in fact, represent a convergence. And it answers some questions right away about photosynthesis, because there's this lone photosynthesizer out here, but this branch created part of the double membranes. It was half of the convergence. The other half came from the actinobacteria. So we still don't know exactly all the details, but we do have an idea about how things may have happened. The single membrane prokaryotes have, as the name says, a single membrane. That photosynthesizer, the single membrane, has the photosystems within the membrane, and if it can move, it has flagellae that wag and that move it around. It's also surrounded by an outside container, which is called a peptidoglycan, and that protects it from this delicate membrane inside from rupturing. The double membrane prokaryotes look exactly the same way, except they have an extra membrane here on the outside. But if they're photosynthetic, they have them here. If they have this inner membrane, it's almost as if the outside, it's almost as if one single membrane prokaryote engulfed another, and then lost its single membrane. So this is an interesting arrangement. You have to ask, what does this do? What's unusual about this? How can this be so successful? Because this double membrane arrangement represents more than 98% of all prokaryotic species. It's really gotten successful because of this coming together, this convergence or this cooperation. Now, what can it do? Well, it makes two compartments for one thing. And we've learned from things like human sanitation that it's nice to have compartments. You know, you don't want to put the sewage in the drinking water. And it's kind of like that with this. We want to keep some compounds here, we want to keep some compounds here, so we can do everything in a more coordinated way. I think that's the reason for the success, and this convergence and cooperation that resulted from the double membranes is really quite impressive. Now, as a result of that convergence, you made the cyanobacteria. These cyanobacteria that are they're called pond scum. This happens to be in a geyser in Nevada. This green scum, those are cyanobacteria. And between 2.7 and 2.4 billion years ago, they had by that time made enough oxygen because they split water, H2O, into hydrogen, which is used biochemically, and then to oxygen, which is released into the atmosphere. And in this process, they started putting their oxygen in the atmosphere. By that long ago, it has started to build up, and it's a toxic waste. Really was. Nobody, no other organisms could use it. But it set the stage by, remember I told you, changing the energy system so that you can get a lot more ATPs, which is the way cells get their energy, from a given amount of food, and maybe increases it by almost an order of magnitude. And once oxygen could be used to increase the yield of ATP, that meant we could have big organisms, because they didn't have to eat as much, and they had less surface, so they didn't need all that surface to get their food, and it meant we could get eukaryotes like us. And now I want to step back to the beginning of genomes and tell you about a problem we had early on. And when we first got our genes, we when we first started looking at genes from sequenced genomes, I told you different genes had different histories. Well, some genes, operational genes we discovered in eukaryotes, have the history that they are, well, they're in eukaryotes. They're a different type of genes. They've come from the double membrane prokaryotes. And furthermore, these double membrane prokaryotes, sorry, I got a little distracted there. The double membrane prokaryotes are, those operational genes make enzymes, and they make functional things that are used, like peptidoglycans, like different molecules used within the cell. And on the other hand, there's informational genes like DNA and RNA. DNA and RNA, they process the information molecules, and they facilitate protein synthesis, the making of proteins. So two very different types of genes. They tended to be operational genes found in eukaryotes were found in Eubacteria, and then another group, the eocytes, had informational genes. Well, within the last few years, we've started to understand this better. So we did again the presence absences of genes. We found out eukaryotes. So let me explain. I talked about eukaryotes, I talked about prokaryotes. Now we're going to talk about the origin of eukaryotes from prokaryotes. We realized eocytes contributed genes to the eukaryotes in this ring. Proteobacteria and the Cyanobacteria contributed other genes to the eukaryotes. So this is really quite a, it's starting now to make sense. It looks as if there must have also been another example of coming together of fusion to make the first eukaryote. We don't know the ancestors and so forth, but now the summary is we know all the organisms though. We had the methanogens, the double membrane prokaryotes, firmicutes, and actinobacteria, and then we have the oldest eukaryotes, is represented by an oldest Charles Darwin, and then we have this group, the eocytes. And these turn out to be extremely thermophilic organisms for the most part. They're sulfur, they eat sulfur, they breathe hydrogen, they live at, some of them can live at above boiling temperature. My wife calls them machocytes. So, talking about machocytes, we come to the next part, and that is where life may have started. And this is not so much our research, but it's to, but I wanted to just take all bases and talk to you about this. Some people have proposed that life started on Earth early in hydrothermal vents. And these vents are quite remarkable in the Pacific. They're at very deep. Temperatures are 300 Celsius, so that's about 700 Fahrenheit. I mean, this is really hot. You say, well, why doesn't this just boil away? But it's so deep, the pressure is so much that the water doesn't boil at 700. But adjacent to it, of course, is water, which is very, very nearly freezing. So we have this incredible source of contrasts of temperature, and we have total contrasts of chemical contrast, totally different chemistry in this water and this water. It's an ideal place with all the energy gradients and all the gradients for life to have started. But there are some alternative sites that have lately been discovered. One is the Lost City, which is a vent off of Florida, about, I don't know, 1,500 miles in the Atlantic. And it has much lower temperatures, 150 to 170, so it's not really boiling. And at the same time, it's alkaline, so whereas these are extremely acidic. And the alkaline is a much more people who do prebiotic chemistry much prefer the alkaline sites. So let's now move on to the last part of this, and I'd like to talk to you about Mars, because I'm a member of the Astrobiology Institute of NASA, and I'm interested in what's going on in Mars. Well, first question to ask is there life on Mars? Or is there water? If we want to know if there's life, we'd like to find the evidence for water. Well, we sent up about 6 or 7, maybe, I don't know how many years ago now, 6 maybe, 2 rovers. Opportunity is one and Spirit is the other. Now, they were supposed to search around on Mars and test things for which minerals are present. Could the minerals tell us there was water present? I think they were supposed to last 90 days, their guarantee. Maybe it's 180, I'm not sure. But up here, this is on day 1600, so that's in Martian days, which are about like Earth days. So that's a long time. Opportunity now is probably up to about 2300. I haven't checked it. But, and Spirit died about a year ago during the winter. It just couldn't get the right orientation to keep enough sunlight to keep alive during the winter. But, you know, for government work, that's pretty good. Anyway, then we put down another lander called Phoenix. And that was really a remarkable one. Phoenix went, I think maybe 4 years ago, 3.5 years ago, and landed on Mars in the Arctic plain during the summer when it was warming up, and it probably only 70 below, who knows, but we also wanted to know exactly where Phoenix was, and it turned out that these high-rise images are taken from a camera in orbit around Mars. Now, this is no ordinary camera. I mean, it must have you talk about it, you sort of think of a little thing, like it's got a lens about like this. I mean, it can resolve from high in orbit, you know, hundreds of miles away to grapefruit. So it's pretty remarkable. And they said, well, we don't know exactly where it's going to land. And they said, hey, you know what? We have high-rise. Maybe we could take a big area, a big photograph, like 50 miles by 50 miles, and then shoot it just as Phoenix is coming down. And they did that. And then they started searching the photograph. And they found out that there were 3 white blips. This is the one that is Phoenix. There's the parachute. There are the lines down to it. And there is the lander Phoenix. Quite amazing. So now we have something on Mars. We had something on Mars which lasts, which we know its location to within a foot on the surface of Mars because we could follow it down. Really quite remarkable. Anyway, more remarkable was what happened, though. When we got there, started digging into the ground, looking for things, it found this white stuff up here are minerals which have been deposited, so don't worry so much about that. But if you can look carefully, there's sort of 3 things down here, which are like 3 little ice cubes. Okay? This is on day 20. By day 24, they had evaporated. They were gone. So water was proven very near the surface. So we're talking about maybe within 3 inches of the surface coming down. This is 2/3 of an inch. And water doesn't melt on Mars. There's too little atmosphere. So under those conditions, water evaporates just, you know, as ice supposed to melt, it in turn evaporates. But we now know that there is water on Mars. There are also clouds on Mars, and the clouds change with the times. They basically, as the South Pole is warming up, it releases water, which gets frozen, travels across the Mars and gets frozen on the North Pole. When the North Pole melts, it moves again and goes to the South Pole. So we know a lot about Mars. Now the next question is there life? Well, we have one more rover that's now being readied, and it's called Curiosity. This is taken about 3 months ago when it was still at Jet Propulsion Lab. It's now at Cape Canaveral being ready to take off. Curiosity is going to look at a strange bit of topography. It's a mountain which is about 5 kilometers high, so that's roughly, if I can do the calculation, I don't know, maybe 2 miles high, but it's surrounded by a basin deep in the soil where water may have been. And from, again, this high-rise photos of this start to show various layers all the way around. And so that's where Curiosity is going to land. It's got a mass spec in it, all sorts of instruments that are going to look for the molecules of life. It's going to dig into those rocks and try to find it, but not just that, at the very earliest phases of Martian evolution. So Laura and I have been invited to see it. We're going to be in Florida the day after Thanksgiving for a launch. We don't know if it'll start, and we've booked 2 extra days, but we're looking for, in case we missed it. You never know for sure, but we're going to see what happens. And so I want to summarize a little bit about cooperation and survival of the fittest. I hope I've shown you that there's a new wave in evolution. It's just not entirely about the harsh realities of life, about the survival of the fittest. It's also about cooperation. But I don't want to give you the feeling that cooperation isn't enforced sometimes. I mean, the mitochondria within us that are making us live and exist and breathe this terrible toxic oxygen are they're probably, we're taking screaming and yelling there, but their net result is they've been able to extend their existence for very long time periods. So we're changing our understanding of the evolution of life on Earth. And I wanted to mention 3 people who have been really important in my career. Okay, there are 3 people who are no longer with us. They are Colin Patterson. Colin Patterson was head of the paleontology department at the Natural History Museum in London. And he was the person who introduced cladistics into the UK and pretty much in part ways into the US, which was a very analytical way of thinking about evolution. And I think also we can answer the question about Colin. I think his empty glass was wine. Another person is Alan Wilson, who's probably the most imaginative scientist I've ever known in my life. Alan was a person who, first when he realized you could sequence DNA, he obtained placentas from people all over the world, because placentas, the mitochondria are inherited through the female side. So he showed that we are all related to a woman who lived in Africa about 200,000 years ago. This is just amazing to me. And he took a lot of abuse for that. But like all of these things, it turned out that he was right. And Alan is no longer with us, but he's absolute force in science. And speaking of this abuse, science is that way. This is the way things get fought out. The new animal phylogeny really took a very long time. Initially, people were yelling and screaming, and everybody had different reasons why it was wrong. But it took until that was started probably in 1995. The crucial paper was published. And it took 10 years for people to come around, and it was only in 2009 when the Royal Society had a meeting to honor the 300th anniversary of Linnaeus. That was incredible. An auditorium of 500 people. Every speaker got up and talked about the new animal phylogeny. And but initially, it was a really big fight, and that's the way science is. By that time, I had sort of said to myself, well, we'll answer the questions, but what you say, how emotional you are, doesn't really make a difference, because the science will decide it, and that's what does it in the end. And then the last person I want to mention is Walter Fitch, who died about 5 months ago. And Walter came to UCLA through WEAG, again, this Wednesday evening group I mentioned before. And we used to have just terrible debates. He was wonderful debates, actually, but very strong. And I would tell him what we were doing. He said, "No, you're doing that all wrong." And then I'd say, "No, it's got to be this and this." He said, "No, no," and then he'd tell me why, and I'd think about it. And next week I'd see Walter, and I'd say, "You know, Walter, you were right, but we have a new result I want to tell you about. Now we really understand this." And he'd say, "No, no, no, you're wrong." And it just, it went on like that maybe 5 or 6 times. And every time I learned so much from him. And I think probably the last one I got him, but I'm not sure of that. Anyway, last, I wanted to thank Laura for putting up with me. Thank you. And I'll be glad to take any questions. And also, I want to thank the audience. You guys were just great. Thank you.
That was a fascinating talk, and thank you for giving it. So in trying to construct a genetic tree that is consistent with what we know from other sources, the more traditional sources, do you think, I wouldn't necessarily expect them to converge with each other, and this is my reasoning. You can take 26 letters in an alphabet, and from that, depending on how you combine them, you could write a manual for a washing machine or War and Peace, right? So with 20,000 or so genes, you don't really need to necessarily see differences between them. It's just the way they're combined that makes them different. The same genes make a caterpillar as a butterfly. So, where are the mutations just kind of a marker for, mutations that have randomly occurred, but they don't really explain the differences in the tree of life? So that's my question.
Okay. That's a very good question. I think what you're referring to is the fact that some mutations are random, and in fact, a lot of them are random. Not all of them are the lucky things that converts an enzyme to something that does something else or changes a gene. Absolutely. So we're using these. Sometimes it does, though, and those are very memorable circumstances. But we're using the genes as a clock. So we say on average a gene will change, a nucleotide will change every so many years. And we just are counting them, and we're using the timing to tell us what the trees look like. And then we're using what we know about the biology to guess what's happening. Okay, great.
Okay. Let's see, we have a question here on the aisle. Just wait 1 second for a microphone, please.
Have we ever found any horizontal gene transfer in single-celled eukaryotes? Or is all the convergent evolution just going on?
Oh, no. Well, I'm certain, I won't say for sure, but I'm reasonably certain that yes, we have found them. In fact, in single-cell organisms, we found some really incredible things. We found something called nucleomorphs, which are entire in the dinoflagellates, which make the ocean colored and so forth. We find that some dinoflagellates, which used to be photosynthetic, lost their chloroplasts, and in return they have swallowed an entire photosynthetic eukaryotic cell. And they've kept the chloroplasts, and they had to keep a little bit of the nucleus, which is called the nucleomorph. But these little little eukaryotes can swallow other eukaryotes too. So the whole process is going on a lot.
Great, thank you. We have a question here toward the center of this row. If we can
Right,
get a
I don't want to answer questions from this guy. Okay, I will.
We'll call security if he gets out of line.
Professor Lake, that was a wonderful lecture. Let me give you a softball question, but it is something that everyone in this audience would be interested in hearing you talk about. We're spending a lot of money. We're going to Mars. You're an astrobiologist. You're interested in the possible existence of life elsewhere. Tell us, please, oh great sir, are we going to find anything? And if so, let's assume we do, why will it matter?
And you called that a soft question. Could I take your hard question instead? No, I think the first thing, why does it matter? It's always this question of curiosity. We don't know what's going to happen. Watson and Crick didn't know what was going to happen when they thought we'll take X-rays of this silly molecule DNA. I mean, science is not predictable, and when you try to predict it, it turns into sort of business as opposed to the exploration. And I think truly important discoveries get made that way. Discovery of the new world, you know, we couldn't, why would you want to go to a new world? There's no gold there. I mean, there's no, you know, but it happened.
Right. Thank you.
Thanks, Bill.
We have a question back here toward the aisle.
Hi, question here. So I have a question about your idea of this convergence. So we usually had two arrows coming together, but if you think about origin of eukaryotes, it seems like we have the mitochondria, so that was one arrow. But then you said that our regular genes come from either Eubacteria or oocytes. So does that mean...
No, not the regular. The operational genes come from Eubacteria, or Cyanobacteria. So it could be the organelles bringing that in. We don't know, but there could be other endosymbioses involved.
So you think there may have been more than one convergence between two?
I think there may have been. I think the history is going to be very complex and very exciting.
Great, thank you. If we can get a question here on the next aisle. Thank you.
May I ask if your remarkable insights into the past allow you to project on future trends in evolution?
I'm sorry, you absolutely never know what's going to happen. I mean, in this business you say, oh, I got it wrong so many times that, but I think that's the excitement of it is you never know what will happen. And yes, when we get things wrong, we acknowledge it. And you go on and you say, well, does this lead us to do something else better? So I just love the excitement of it.
Actually, just a follow-up to that, Jim, if I could ask, what is the evidence that evolution continues to occur?
Well, that's very good. We have a lot of experiments that have been done. There's an experiment that's been done at Michigan State where they've taken a bacterium and they've grown the bacterium they've done very long-term studies. So they grew this initial E. coli bacterium into, I don't know, maybe 8 or 16 different groups. And then they followed the evolution of these groups and the evolution within them and so forth. And you can just see the DNA change with time. And some of these bacteria change, some of the proteins change. So this has been all observed in the laboratory. So.
Wonderful, thank you. Okay. We have a question down here toward the front on the aisle.
Thank you. That was a very interesting lecture. I recently read an article that 90% of all human DNA is based on microorganisms. And yet if we all evolved from microorganisms, why wouldn't it have been 100%, or is there a time in the transition that there was human DNA?
Well, I haven't read the article, so I don't know what the article could say. I mean, I could think of lots of things, but if it's 90%, you know, sometimes you can't tell the difference between 90% and 100%. It's like, these are all statistical questions, and you really have to, that's one lesson that I really didn't understand until I got into evolution, but my training has been as a physicist. Because I wanted, before my brain froze, I knew I wanted to be a biologist. Before I started purifying RNAs and things, I took all these, you know, graduate courses in quantum field theory and general relativity, all those things. And then I realized, oh my God, I should have also been taking courses in statistics, because everything gets proven by numbers. And statistics is such a subtle, beautiful science, and I don't know enough of it, but I just see that as part of a lack in our scientific education. But it's really powerful, really powerful.
One toward the center there, yes, please.
Yes, thank you very much for the lecture. Referencing the double membrane.
I don't see where you are.
Oh, I'm sorry.
Oh, okay.
Yeah.
Okay, you're behind somebody.
Referencing the double membrane.
Sure.
On the mitochondrion and also the chloroplast potentially having been, or almost for sure having been, organisms that were incorporated. Can you speak toward the nucleus and its double membrane, or is that different enough that there was no chance that that was at one point an organism that was incorporated?
Well, that's, you know, this is almost a planted question, I think, but it's not. I personally think that the nucleus is an endosymbiont, and that's when we started, we have actually published several papers on it, maybe 3 or 4, in some high visibility publications like Nature and Science. So that's an idea that's out there. That idea seemed to be going nowhere till about 5 years ago, and within the last 3 years there have been a number of papers written about that very idea, supporting it because the details of it depended upon being able to go back very early in time. And just to make a short answer, Science had a wonderful article about it, about this work of other people who have started to prove this, and it's sort of feeling to me a little bit like when the new animal phylogeny came out. I think the world is now starting to be prepared for that. So that's wonderful that you came up with that idea too, and I wish more people would. Okay, thank you all.
Videocassette copies of this program are available for purchase from the UCLA Instructional Media Library. Call toll-free 1-877-958-2200. Additional information about the people, places, and ideas discussed in this program is available at our website, www.webcast.ucla.edu.